Today we are going to the source. We are unpacking chapter 54, and we are going to look at the mechanics of this war.
And that word mechanics is so key here. The source material for this deep dive is fascinating because it strips away the magic. It doesn't treat the immune system as this vague force of nature. It treats it as biochemistry.
Yes.
It treats it as engineering.
Which I love, because when you actually look at the details, the nuts and bolts of how these cells work, it is mind-blowing. We are talking about cells that can shapeshift to walk through solid walls. We're talking about communication networks that are more sophisticated than the internet. And, as we will find out, we are talking about the fact that your body literally manufactures household bleach to kill its enemies.
It really is a biological battlefield. And like any army, to understand it, you have to understand the soldiers, the weapons, and the logistics.
So let's start with the mission. The source material sets the stage beautifully. We are constantly exposed to bacteria, fungi, viruses, parasites. We are walking petri dishes. So survival depends on this army, the leukocytes. But this isn't a clone army, is it?
No, absolutely not. If you look at the roster provided in the text, you see these distinct classes of soldiers. It's exactly like a modern military. You wouldn't send a fighter pilot to clear a building, and you wouldn't send a medic to fly the jet. You need specialization.
So walk us through the lineup. Who are the main players here?
The first group we have to talk about are the neutrophils. If we are sticking with that military analogy, these are your Marines, your heavy infantry.
Why do you call them the heavy infantry? What makes them the grunts on the front line?
Because of the sheer numbers. The text notes that neutrophils make up about, 60% of all the white blood cells circulating in your body? They are by far the most abundant.
60%, yeah.
And they are specialists in one thing, phagocytosis.
Phagocytosis. That's a great word. Let's break that down.
It comes from the Greek phagein, meaning to eat. These cells are quite literally eaters. Their primary job is to rush to the front lines, usually a bacterial or fungal infection, grab the invader, and physically ingest it.
So they eat the enemy alive?
They eat the enemy alive. It's brutal and incredibly effective.
Simple, brutal, effective. But they aren't the only ones doing the eating, right? The text mentions monocytes as well.
Right. Now, monocytes are fascinating because they are the, let's call them the transformers of the immune system. When they are floating in your blood, they are just monocytes. They are relatively quiet, just circulating.
Yeah.
But when they get the signal to leave the blood and enter the tissue, they undergo this incredible metamorphosis.
They change, like physically change.
They physically and chemically differentiate into something called macrophages.
Which, if I remember my Greek, literally translates to big eaters.
Exactly, and the name fits. Macrophages are larger than neutrophils, and they are much longer lived. While a neutrophil might rush in, kill a few things and burn out, the macrophage settles in. They are the sentries.
The cleanup crew.
The cleanup crew and the guards, they patrol your tissues even when you aren't sick, eating debris, eating dead cells, and just generally keeping the area clean.
So neutrophils are the shock troops, macrophages are the heavy tanks, or the garrison.
That's a great way to put it.
What about the specialists?
Yeah.
I've always been confused by eosinophils. They sound exotic.
Eosinophils are your heavy artillery, and they're designed for a very specific type of enemy, the parasite.
Like a tapeworm or something.
Exactly. And I want you to think about the scale problem here. A bacterium is tiny. A single neutrophil can eat it, no problem.
Yeah.
But a parasitic worm, that is massive compared to a cell.
Righty.
A white blood cell trying to eat a worm is like you trying to eat a telephone pole. You can't do it. It's physically impossible.
So what do? They can't just ignore it.
They swarm it. Eosinophils attach to the surface of the parasite and release their granule, these little packets of toxins, to basically burn it from the outside in. They just bombard it until it dies.
That is incredibly aggressive.
It has to be. And then you have the basophils. These are the rarest of the bunch, but they play a crucial role as the alarm system.
The alarm system.
They are packed with granules containing signaling molecules, specifically things like histamine.
Histamine. That's the stuff that makes me sneeze during allergy season.
It is. But in a real infection, that histamine is the siren. It screams, "We're under attack," at a chemical level and triggers inflammation to bring in the rest of the troops.
Okay, so that's the innate response. What about the lymphocytes? They feel like a whole other category.
They are. The lymphocytes are the special forces and the intelligence officers. This includes your B cells, T cells, and natural killer cells. We are going to touch on them later in the discussion because they represent the adaptive immune system, the part that learns and remembers.
The institutional memory of the army.
Precisely. But for the bulk of this chapter, we are focusing on that first line of defense, the innate system, the eaters and the chemically armed soldiers.
Okay, so we have the roster. We have the neutrophils, monocytes, eosinophils, and basophils. But here's something that stood out to me in the source material. The turnover rate. These cells don't live forever.
Don't live forever is a massive understatement. The text highlights a concept called hematopoiesis, the production of blood cells. And it notes that the myeloid cells, the neutrophils, and their cousins live only a few hours to a few days in circulation.
Wait, hold on. Hours?
Yes. For a neutrophil in the bloodstream, its lifespan can be as short as 6 to 10 hours.
So the neutrophil guarding my left toe right now might be dead by lunchtime.
Correct. It's entirely possible.
That seems wildly inefficient. If they die that fast, how on earth do we keep an army staffed? We must be making them constantly.
We are. It is a massive, ceaseless industrial effort happening inside your bone marrow. This is one of the most chemically regulated supply chains in nature.
Let's dig into that supply chain aspect, because the text mentions a lot of specific proteins here. Growth factors.
Right. Imagine the bone marrow as a factory. It has the raw materials, these pluripotent stem cells. These are the progenitors that can become anything. But the factory needs to know what to build. It needs purchase orders.
Okay, I like that analogy.
Yes.
Who sends the purchase order?
The tissues, the infection itself. The text lists specific molecules. Stem cell growth factor, which gets things started, and something called GM-CSF.
GM-CSF. That sounds like a car company part number.
It's not far off. It stands for granulocyte macrophage colony stimulating factor.
Okay, so it does exactly what it says.
It does exactly what it says. It stimulates the growth of a colony of granulocytes, which are mainly neutrophils and macrophages.
So let's say I get a cut on my finger, bacteria gets in, my body realizes it needs more soldiers. It releases this GM-CSF.
Exactly. And it travels through the blood to the bone marrow and essentially hits the override button on the production line. It says, stop prioritizing red blood cells for a minute. We need infantry. Shift production to neutrophils now.
That is incredible. It's dynamic resource allocation in real time.
And it gets even more specific. The text mentions interleukins, like IL-5.
Interleukin-5, okay.
If you have a parasitic infection, your body doesn't just send out a general call to arms, it releases IL-5. Why? Because IL-5 specifically tells the marrow to make eosinophils. It doesn't waste energy making neutrophils that can't kill the worm. It orders the specific weapon needed for the specific enemy.
So the body isn't just panicking, it's strategizing its manufacturing based on the threat.
Precisely. It's an incredibly intelligent system.
Okay, so we've built the soldiers, billions of them. They're flooding out of the bone marrow and into the bloodstream. But now we hit a logistical problem that the source material spends a lot of time on, the commute.
Right. This is a huge challenge because the soldiers are in the blood, but the infection is almost always in the tissue outside the blood vessel.
It's like the army is on the highway, but the battle is in the houses 3 blocks away.
That's a perfect analogy. And the blood is moving fast. So the cell has a 3-part challenge. 1, it has to know where to get off the highway. 2, it has to physically stop moving in a rushing river of blood. And 3, it has to push through the wall of the blood vessel to get to the tissue.
Let's take those one by one. First, the where. How does a blind cell floating in blood know that there is an infection in my finger? It doesn't have eyes. It can't see the exit 54 sign.
It uses chemotaxis.
Chemotaxis.
This is biochemical sensing. The source describes it as movement in response to a chemical gradient. Think of it like a scent trail.
Scent trail?
Yeah.
Like a shark smelling blood from a mile away?
It's a very similar concept, but on a microscopic scale. The infection site is releasing chemicals. These can be bits of the bacteria itself, like their peptides or chemokine signals sent out by your own damaged cells screaming for help. These chemicals diffuse out into the blood.
So the concentration is highest at the cut and gets lower and lower the further away you get.
Right. The neutrophil has receptors on its surface that can detect these chemicals. But here's where we need to get into the weeds of the mechanism, because this isn't magic. The text mentions G-protein coupled receptors.
GPCRs. I remember seeing that diagram. It looks like a snake weaving in and out of the cell membrane.
That's the one. It passes through the membrane 7 times. This is the sensor. When a chemokine molecule floats by and hits that receptor, it clicks into place, like a key in a lock.
And that turns the key.
It turns the key. And this triggers a cascade inside the cell. The text specifies that it activates an enzyme called phospholipase C.
Okay, phospholipase C. What is its job in this cascade?
Its job is to release the trigger mechanism, calcium. It causes the cell to flood its own cytoplasm with calcium ions, K2+.
Calcium. That's what muscles use to contract, right? I feel like I remember that from biology.
Exactly. And that is not a coincidence. The white blood cell isn't a muscle, but it has a skeleton, a cytoskeleton made of actin and myosin, just like your biceps. When that calcium hits, it activates the actin and myosin.
So the smell of the bacteria literally causes the cell to flex.
It causes the cell to crawl. The actin polymerizes, it links together rapidly, pushing the cell membrane forward in the direction of the smell. It builds what's called a pseudopod, a false foot.
False foot.
Then the myosin contracts at the back of the cell to pull the rear forward. It's this continuous process of reaching and pulling, always moving towards the stronger signal.
It's like a tank that builds its own treads in front of it as it drives. That's incredible.
That is a perfect analogy. It is a self-building machine. It crawls toward the strongest signal.
That is mind-bending. But wait, it's crawling inside the blood vessel. The blood is rushing by. If it just starts crawling, won't it just get washed away?
It would. Which brings us to the second challenge, the stop, or as the chapter calls it, adhesion.
This is the Velcro part.
It is molecular Velcro, but it's a two-stage process, which is really clever. The text distinguishes between two families of adhesion molecules, selectins and integrins.
Selectins and integrins. What's the difference?
Think of selectins as the braking system. They are on the surface of the blood vessel wall, the endothelium. As the neutrophil rushes by, the selectins reach out and grab it. But it's a weak grab, a low-affinity interaction.
So it doesn't stop the cell dead in its tracks.
No, it just slows it down. The cell starts to tumble. It rolls along the wall of the vessel. The text really calls this rolling adhesion.
Like a tumbleweed catching on thorns in a fence.
Exactly. But to stop, to really anchor itself so it can crawl out, it needs the heavy-duty clamps. Those are the integrins.
Now, the outline highlights the structure of these integrins. It mentions alpha and beta subunits. Why is that specific detail important?
Because integrins are complex machines. They consist of two different protein chains, an alpha chain and a beta chain, that are non-covalently linked. They work together to form the grabber. Specifically, the text focuses on the beta-2 subunit, which is also known as CD18.
CD18. Okay, I'll try to remember that.
Keep that name in mind. These integrins are on the white blood cell. When the cell detects that chemokine signal we talked about, it's not just about movement. That signal also tells the integrins to snap open. They activate.
So the smell of the infection turns on the superglue.
Basically, yes. And they lock onto proteins on the vessel wall called ICAMs.
Intercellular adhesion molecules.
Yes. Specifically, the text points out that an integrin called LFA1 binds to ICAM1 on the wall, and this bond is tight. The rolling stops. The cell is now stuck fast to the wall right next to the infection site.
So selectins slow it down, integrins lock it down.
Correct. It's a two-step process to make sure it stops in exactly the right place.
But what happens if that lock is broken? The source material mentions a specific disease. Type 1 leukocyte adhesion deficiency.
This is a heartbreaking condition, but it teaches us so much about how the system works. In this disease, the patient has a genetic mutation. They cannot make that beta-2 subunit, the CD18 we just talked about.
So they are missing a critical piece of the clamp.
They are missing half of the heavy-duty clamp. The LFA1 integrin can't form properly. They're missing the superglue.
So what does that look like for the patient? What happens?
Well, imagine the scenario. You have an infection. Your bone marrow does its job. It pumps out billions of neutrophils. The army is there. It rushes to the scene in the bloodstream. The selectins grab them and they start to roll.
That they can't stop. They can't stick.
They can't stop. They roll right past the battlefield. They just get washed away in the blood. So the patient suffers from these severe recurrent bacterial and fungal infections. They have the soldiers, but they can't deploy them to the battle.
That is just tragic. It really highlights that having the cells isn't enough. The mechanics of getting them there are just as critical.
Absolutely. No logistics, no victory. It's a fundamental rule of warfare, even at this scale.
So, assuming the machinery works, the cell has stopped, it's anchored, now it has to get through the wall, the diapedesis.
This is the third and final challenge of the commute. The blood vessel is lined with endothelial cells. They're like paving stones fitted tightly together. The neutrophil has to squeeze between them without causing a leak.
And we are talking about a physical squeeze here. This isn't a chemical trick.
A very tight squeeze. The text references figure 54.2, which shows this beautifully. The neutrophil inserts that pseudopod, that false foot, into the tiny gap between two endothelial cells, and then it pushes.
It wedges the door open.
essentially. And this is where the shape of the cell itself, specifically its nucleus, becomes incredibly important. You know how most cells in a textbook drawing have a nice, big, round nucleus, like a ball in the center?
Sure.
Neutrophils are different. Their nucleus is multi-lobed. It looks like a few sausage links connected by a thin string.
Why? What's the advantage of that?
For this exact moment, a big, round ball is hard to squeeze through a crack. It's rigid. But a series of smaller, linked sausages, that's flexible. It allows the nucleus to deform and snake through the gap, one lobe at a time.
Form follows function.
Always. The cell pulls itself through the wall and emerges into the tissue. It has arrived at the battlefield.
Okay, that whole journey is just astounding.
Yeah.
But let's circle back for a second. We talked about the signal, the chemokines. This brings us to one of the most technical but surprisingly crucial parts of the source, the structure of the signal itself.
Yes. The language of the immune system.
The text goes into detail about disulfide bonds and CC versus CXC subfamilies. I have to admit, my eyes glazed over a bit at first. Why does the spacing of these amino acids matter so much?
It matters because it's the code. Think about a protein as a long string of amino acids, but to work, it has to fold up into a very specific 3D shape.
Like protein origami.
Exactly like origami. Now, these chemokines are small. They are tiny proteins floating in a chaotic, hot, watery environment. To keep their shape, to not fall apart, they're stapled together.
Stapled.
Chemically stapled. That's what a disulfide bond is. It's a strong covalent bridge between two cysteine amino acid residues, sulfur to sulfur. It locks the loops of the protein in place so it doesn't unravel.
Okay, that makes sense. It's for stability. But what about the CC versus CXC thing? That seems so minor.
That refers to the position of the first two cysteines, the first two staples. In the CC family, the two cysteines are right next to each other in the protein chain. In the CXC family, there is another amino acid, the X, separating them.
That seems like a tiny difference. One amino acid.
Structurally, it's huge. It completely changes the shape of the protein loop, which changes the overall shape of the chemokine. It changes the shape of the key. And because the shape of the key changes, the lock it fits into the receptor in the white blood cell has to be different. The text notes that CXC chemokines, like IL-8, typically attract neutrophils. CC chemokines, on the other hand, typically attract monocytes and other cell types.
Simply by adding one spacer amino acid, the body changes the message from call the Marines to call the cleanup crew.
Exactly. It is a highly specific and elegant molecular code. The language is built into the structure.
Okay, so the code was sent. The Marines, the neutrophils, have arrived. They have crawled to the bacteria. Now, it's time for violence. We are entering part 3, the attack.
This is where the biochemistry gets really intense. We are talking about the mechanics of killing.
So the cell touches the bacteria. Does it just eat it immediately? How does it grab on?
It can, but it prefers if the food is prepared first. The process is called opsonization.
Opsonization. I've heard this described as buttering the toast to make it easier to eat.
That's a good way to put it. Opsonins are molecules that coat the bacteria to make them tastier or easier to grab. These can be antibodies, which we'll touch on later, or something called complement factors. They act like little handles. The neutrophil has receptors that specifically grab these handles, which makes the whole process of enveloping the bacteria and pulling it inside much more efficient.
So the bacteria is now trapped in a bubble inside the cell, a phagosome.
Right, but it's still alive in there. Now the execution begins. The phagosome fuses with granules, these bags of enzymes and chemicals, inside the cell, and this triggers the main event, the respiratory burst.
Respiratory burst. It sounds like the cell is taking a deep breath before the fight.
In a way, it is. The cell's oxygen consumption spikes massively, up to 50 times its normal rate. But it isn't using that oxygen for energy in the normal way, it's weaponizing it.
This is the part I really want to drill down into because the chemistry is wild. We are talking about the NADPH oxidase system.
Yes, this is the engine of destruction. NADPH oxidase is a massive enzyme complex that assembles on the wall of that bubble, the phagosome. Think of it like a high-voltage transformer.
Okay, a transformer, what is it moving?
Electrons. It takes high-energy electrons from a molecule called NADPH, which is basically a cellular battery, and forcibly attaches them to molecular oxygen O2.
And oxygen doesn't like that.
Oxygen hates being unstable. Its stable is O2. When you add that extra electron, it becomes superoxide O2 with a negative charge and a dot.
A free radical.
A very potent and dangerous free radical. It's chemically reactive and will tear apart any biological molecule it touches, but that is just step one. The text shows the equation. 2O2 plus NABPOH gives you 2 superoxides plus NADP plus and a proton.
So we've made superoxide inside the bubble, but the source says it doesn't stop there.
No, superoxide is just the starting material for the really nasty stuff. It spontaneously dismutates, it reacts with itself to form hydrogen peroxide, H2O2.
Hydrogen peroxide. That's literally the stuff in the brown bottle in my bathroom cabinet that you put on cuts.
The very same. So now, inside this microscopic bubble, the cell is dousing the trapped bacteria in hydrogen peroxide.
Which is bad enough for the bacteria, I imagine. But then comes the bleach. This part is unbelievable.
Then comes the bleach. This is the part that still amazes me every time I read it. The granules in the neutrophil contain an enzyme with a green color called myeloperoxidase.
Myeloperoxidase.
This enzyme grabs the hydrogen peroxide that was just made. Then it grabs a chloride ion, Cl, which is just floating around everywhere from the salt in your body, and it smashes them together.
Peroxide plus chloride.
Creates hypolous acid, HOCl.
Which is, what, chemically?
It's hypochlorite. It's the active ingredient in household bleach.
So just to be absolutely clear, my white blood cells are actively manufacturing Clorox inside my body to kill bacteria.
Yes. They are chemical factories producing industrial-grade disinfectant at point-blank range, sealed inside a little bubble with the enemy.
That is absolutely terrifying and amazing in equal measure.
And here is a fun fact from the text that just connects everything. Myeloperoxidase, that enzyme that makes the bleach, contains a heme group, similar to hemoglobin. But instead of being red, it's green.
Green.
Yes. This enzyme is so incredibly abundant in neutrophils that it's what gives pus its characteristic green color.
Wait, no, you're kidding. So when a cut gets infected and there's that greenish pus, that color is literally the enzyme.
That color is the massive accumulation of the bleach-making machine.
Yeah.
It's visual proof that your army is there in huge numbers fighting the chemical war.
I will never look at an infection the same way again. It's not just growth, it's a chemical stockpile.
Exactly, a battlefield.
But the source lists other weapons too.
Yeah.
It's not just oxidative stress, right?
Yeah.
Because what if the bacteria is resistant to bleach for some reason?
A great question. Then you hit them with the non-oxidative arsenal. Table 54-2 in the source lists these out. You have lysozyme.
What does lysozyme do?
It hydrolyzes bacterial cell walls. It basically dissolves their armor. It specifically breaks the chemical bonds holding their peptidoglycan shell together.
And defensins. The name sounds pretty straightforward.
Defensins are like little daggers. They are small antibiotic peptides that are positively charged, and they are attracted to the negatively charged bacterial membrane. They insert themselves and punch physical holes in the membrane, causing the bacteria's guts to leak out.
And what about lactoferrin? That sounds different.
This is a strategy of starvation. Bacteria, like all living things, need iron to grow and divide. Lactoferrin is an incredibly powerful iron-binding protein. The neutrophil dumps it into the phagosome to mop up every last free iron atom.
So it starves them of a key nutrient.
While the bleach burns them, the lysozyme dissolves their armor, and the defensins are poking holes in them. It is a multi-pronged attack designed to ensure absolutely nothing survives.
It sounds foolproof, but biology is never foolproof. The source material discusses what happens when this incredible weapon system fails. Chronic granulomatous disease, or CGD.
This is the flip side of the coin, and it shows just how important that respiratory burst is. In CGD, there is a genetic mutation in one of the genes that build that NADPH oxidase transformer we talked about.
So the electron pump is broken.
Correct. The cell can move just fine. It can perform chemotaxis, it can adhere, it can even eat the bacteria the phagosome forms, but it can't generate the superoxide. It can't make the bleach.
So the bacteria is sitting inside the cell, safe and sound, in a little protective bubble.
Exactly. The neutrophil swallows it, but can't kill it. In fact, for some bacteria, the neutrophil becomes a perfect incubator.
That is a nightmare scenario.
It leads to severe, persistent infections. But notice the name, chronic granulomatous disease. Since the body can't kill the invader, it tries to do the next best thing, contain it. It builds a wall of immune cells around the infected cells to trap them.
And that wall is called a granuloma.
Right. A granuloma is basically a prism built by the body for enemies it cannot execute. It's a physical sign of a failed respiratory burst.
It's amazing how the pathology explains the physiology. You break the weapon, You see the direct result. Now, speaking of weapons, we have to talk about the Spider-Man maneuver. This is my favorite part of the chapter.
Ah, yes. Nets. Neutrophil extracellular traps.
When I read this part of the chapter, I thought it had to be science fiction.
It revolutionized our understanding of these cells. For decades, we thought neutrophils only killed by eating things. But what if the invader is too big? We talked about parasites, but what if it's a big clump of fungi, like a hyphal network?
You can't eat something bigger than your head.
Exactly. So the neutrophil performs a final sacrificial act. It casts a net.
But the net is made of itself.
It is made of its own DNA.
Walk us through the mechanics of this, because throwing your own DNA at someone sounds impossible. DNA is packed incredibly tight inside the nucleus. How do you get it out?
Right. It starts with a very specific enzyme called peptidylarginine deaminase, or PAD4.
That's a mouthful.
It is. But its job is very cool and very specific. It goes into the nucleus and attacks the histones.
Histones are the spools that the DNA thread is wrapped around, right?
Yeah.
To keep it organized.
Yes. DNA is negatively charged. Histones have positively charged amino acids, like arginine. Opposites attract, so the DNA wraps tight. This enzyme, PAD4, citrullonates the histones. It chemically converts the positive arginine into a neutral amino acid called citrulline.
And so the charge disappears.
The positive charge is gone. Suddenly the magnetic attraction is broken. The DNA lets go of the spool. It decondenses. It unwinds and expands explosively inside the cell.
So the nucleus just swells up and pops.
It ruptures. The decondensed chromatin, the DNA, fills the entire cell. At the same time, those granules, the bags of bleach-making enzyme and defensins, rupture too. So now the sticky DNA strands are coated in poison.
And then, what's the final step?
The cell membrane bursts, the neutrophil dies, but as it dies, it ejects this massive, sticky, toxic web of its own DNA out into the tissue.
Like Spider-Man shooting a web to catch a villain.
It's the perfect analogy. It traps the bacteria or fungi physically. They get stuck in the DNA strands, and because the strands are coated in those antimicrobial enzymes, they get killed and digested right there in the net.
It's a suicide mission. The soldier blows himself up to stop the enemy advance.
It is one of the most heroic and simultaneously grotesque mechanisms in the human body.
This brings up a darker side of this whole military operation.
Yes.
Collateral damage.
Yes, friendly fire. This is a huge issue.
If you are spraying bleach, throwing toxic nets, and releasing enzymes that dissolve cell walls, surely you aren't just hurting the bacteria, you have to be damaging your own tissue.
You are. These weapons are, by their nature, indiscriminate. The source material spends a significant amount of time discussing the balance between proteases and antiproteinases.
Let's break that down. Proteases are enzymes that chew up protein.
Right. Specifically, the text mentions elastase. This is an enzyme neutrophils release to break down bacterial proteins. But our lungs are also made of a critical protein, specifically elastin.
Elastin is what makes the lungs stretchy. It gives them their recoil so we can breathe out passively.
Correct. So if elastase is released in the lungs during an infection, it has the potential to digest the lung tissue itself. It can turn your spongy, flexible lungs into flabby, useless sacs. This is the fundamental mechanism of emphysema.
But we have neutrophils in our lungs all the time fighting off minor infections. Why don't we all have emphysema?
Because we have a shield. The liver works around the clock, pumping out a protein called alpha-1 antitrypsin.
The antiproteinase.
Yes. Think of it like a custom-fit sheath for the elastase sword. It floats around, finds any free elastase, binds to it, and completely neutralizes it. It stops the friendly fire before it can do any damage.
So there is a constant balance. The army has powerful weapons, elastase, but the civilian population has shields, alpha-1 antitrypsin.
Exactly. It's a system of checks and balances. But we can break that shield. The source makes a fascinating and very direct connection to smoking here.
How does smoking affect this delicate balance?
It comes down to a single amino acid in the shield protein. The alpha-1 antitrypsin protein has a key methionine residue in its active site. It needs that specific methionine to grab and neutralize the elastase.
Okay.
Cigarette smoke is loaded with chlorinated oxidants and other reactive species. When you inhale smoke, those chemicals go into your lungs and they attack that methionine. They oxidize it.
Like rusting the key part of the shield.
Effectively, yes. They chemically alter the shape of the shield. So now the alpha-1 antitrypsin cannot bind to the elastase. The sword doesn't fit in the damaged sheath anymore.
So the shield is down.
The shield is down, but the neutrophils are still there because smoking causes inflammation, so they're releasing elastase. And now that elastase is completely free to chew up your delicate lung tissue unchecked year after year.
So smoking causes emphysema not just by, depositing tar in the lungs, but by chemically tricking your own immune system into eating your lungs from the inside out.
Precisely. It destroys the regulation. It tips the balance of the war in favor of self-destruction.
That is a sobering realization. It really shows that health isn't just about fighting the enemy, it's about controlling your own army.
Control is everything. And control requires communication. Which brings us to the final major piece of the puzzle from this chapter. Signaling. The language of the immune system.
The text lists a laundry list of molecules here. Cytokines, histamine, leukotrienes. It's an alphabet soup. Let's try to organize it. We already mentioned histamine, the alarm bell.
Right, histamine is the opener. We saw it with basophils.
The source shows its structure. It's made from the amino acid histidine.
Yes, it's a very simple modification. You just chop off a carboxyl group, a process called decarboxylation, and boom, you have histamine.
What does it actually do to the battlefield? What does opening the gates mean?
It causes vasodilation. It makes the small blood vessels wider and leakier. This is what causes the classic signs of inflammation. Redness from increased blood flow, heat for the same reason, and swelling.
Which we usually think of as bad. We take antihistamines to stop it.
But biologically, it's absolutely necessary. That swelling is the plasma fluid flooding into the war zone. It brings antibodies, it brings complement proteins, and most importantly, it physically opens up the gaps between the endothelial cells to make it easier for the white blood cells to get through. The swelling is the mobilization.
Okay, so histamine opens the gates. What about the radio chatter, the cytokines?
Cytokine is a very broad term for small signaling proteins, but the text focuses on interleukins. The name literally means between leukocytes. It's how white blood cells talk to each other.
Give us an example of a conversation they might have.
Well, we already talked about IL-5 calling for eosinophils from the bone marrow. That's a long-range recruitment order. Then you have cytokines like IL-1 and TNF-alpha. These are the general alarm signals. A macrophage that eats a bacteria will release these, and they travel throughout the body. They go to the brain to cause fever.
So a fever isn't just a random side effect of being sick. It's a direct response to a radio signal sent from, say, my infected finger.
Exactly. The local unit radios command, and command the brain, turns up the thermostat on the whole building to make it harder for the bacteria to reproduce and to make the immune cells work faster.
It's so integrated. And then you have the lipid mediators. The text gets deep into arachidonic acid here, which sounds complicated.
These are the eicosanoids. They're interesting because they aren't stored in granules. They're made on demand from the fats in the cell membrane itself.
The source mentions two big families, leukotrienes and prostaglandins.
Right. Leukotrienes are the heavy hitters for sustaining inflammation and causing smooth muscle contraction. The text notes their structure features conjugated double bonds.
Sick memes.
It creates a rigid, specific shape. These molecules are incredibly potent at constricting airways. This is a key player in what happens during an asthma attack. The immune system thinks it's fighting an invader in the lungs and accidentally closes your throat.
And prostaglandins. I know those are related to pain.
They are. They regulate local blood flow and they sensitize nerve endings to pain. When you take aspirin or ibuprofen, you are blocking the enzyme cyclooxygenase that makes prostaglandins. You are essentially cutting the phone line so the pain message doesn't get sent as strongly.
So we've covered the innate system, the eaters, the bleach, the nets, the signals. But the chapter ends with a nod to the adaptive connection, the lymphocytes.
The special forces, the intelligence division.
We can't do a full deep dive on them today. That's a whole other chapter. But we should define their roles as the text does. We have B cells and T cells.
Right. B cells are the factories for antibodies. This is what's known as humoral immunity.
Humoral, as in the fluids of the body.
Yes. They mature in the bone marrow. There's a fun fact from the text. The B actually comes from the bursa of Fabricius, which is an organ in birds where B cells were first discovered. But in humans, bone marrow works as a perfect mnemonic.
So B cells shoot arrows, the antibodies, from a distance. What about T cells?
T cells mature in the thymus, hence the T. And there are two main types we should mention. Helper T cells are the generals. They don't kill anything themselves. They coordinate the entire attack. They look at the intelligence presented by macrophages and then activate the B cells and the other T cells. They're the cellular switchboard.
Yeah, the other type.
Cytotoxic T cells, the assassins. Their job is to find and kill your own cells that have gone rogue. If a cell has a virus reproducing inside it, or if it's become cancerous, the cytotoxic T cell recognizes it and forces it to commit suicide, a process called apoptosis.
How does it do that?
It punches holes in the target cell with proteins called perforins and then injects granzymes, enzymes that trigger the self-destruct sequence. It's ruthless.
It has to be to stop the infection from spreading.
It has to be.
So, putting it all together, we have looked at the factory in the bone marrow, the incredible commute involving chemotaxis and diabetes, the violent attack with phagocytosis and the respiratory burst, the suicide tactics of nets, and the terrible cost of collateral damage in diseases like emphysema.
It is a massive, violent, elegant, and interconnected system.
What is your biggest takeaway from this deep dive, from really getting into the weeds of this chapter?
For me, it's the realization of what inflammation actually is. We spend so much of our lives trying to stop it. We ice injuries, we take ibuprofen, we eat anti-inflammatory diets, and chronic systemic inflammation is bad. Yes, we know that. But acute local inflammation, that redness and swelling around a cut,
Yeah. What about it?
that is the beautiful, visible evidence of the system working perfectly. That heat is the metabolic heat of the respiratory burst. That swelling is the highway opening up for the troops. That pain is the signal telling you to protect the area. Without inflammation, the army never arrives, and you die from a paper cut.
It redefines the experience of being sick. It's not just a collection of miserable symptoms. It's a tactical response. It's the sound of the battle.
Exactly. It's the price of security.
So to the listener, right now as you sit there listening to this, you are a fortress. You have billions of soldiers patrolling your walls. They are shape-shifting, they are talking to each other, and they are manufacturing ammunition on the fly. You are safe because they are vigilant.
We are walking, talking biological citadels, and the war is constant.
I love that image. Next time you see a bit of greenish pus or feel a fever coming on, don't just be annoyed. Maybe take a moment and salute the troops. They're fighting the good fight for you.
Stay curious and maybe wash your hands. It gives them less work to do.
Thanks for listening to The Deep Dive. See you next time.